Howdy, welcome to Horticulture Innovators, a podcast series where we will explore impactful innovations in horticulture. I'm Amit Dhingra, your host. I'm a professor and head of the Department of Horticultural Sciences at Texas A&M University. In each episode, we'll explore the inspiring stories of the pioneers who are contributing to sustainability, wellness, and food security through their amazing work in horticulture. Welcome to another episode of Horticulture Innovators. And today we have with us Professor Arun Sharma. He's from the Department of Plant Molecular Biology at the University of Delhi South Campus, a place where I got my Ph.D. from, and you're the one who trained me during those first years of my Ph.D. So welcome, Dr. Sharma.
Thank you.
Thank you. So glad to have you here. And today we just want to talk a little bit about your journey, because you've recently retired. So congratulations.
Thank you.
And really want to discuss that and how you got interested in all these things. And so let's start at the beginning. What got you interested in science to begin with?
Yeah, so basically my journey was driven a lot by the kind of teachers I had. And some of the teachers taking certain subjects, they influenced me much more. So in the high school, I had a biology teacher and I was very much interested in his teaching. Yeah. And I decided to pick up biology in later classes also. And then, in the bachelor course, we had 3 subjects like botany, zoology, and chemistry. And I had very nice teacher teaching botany, and he was my favorite teacher. And then I decided to do masters in botany. And again, similar situation there. I had a teacher who was taking biochemistry and physiology course, and he taught phytochrome work very well. And so when I joined my PhD program at JNU, and Professor Sopory, my mentor, he was also working on that area related to phytochrome. So I ended up working on phytochrome control of nitrate assimilation there.
Wow.
So this is something which, like many people say, teachers have, great influence on students. That's what it was for me.
And I think you passed it on, because you had a great influence on me as well, because I remember when I was starting to do my PhD, how patiently you taught things. And the intellectual curiosity was, enhanced every time we were discussing things. But before we move forward, I just wanted to say phytochrome is basically the protein that senses red and far red light in plants, just like we are able to see light with our photoreceptors in our eyes. That's what it is. So that's pretty cool. So, and you also said nitrate reductase. So what does that do? What's its significance in plants?
Yeah, it's one of the very important enzymes for nitrate assimilation, which leads into synthesis of amino acid, and ultimately they go into protein. So there are some studies which indicate, they compare different cultivars, low yielding, high yielding, medium yielding, and they find some correlation between activity of nitrate reductase and high yield, like high the activity of enzyme and high yield. So it's kind of enzyme related to nitrogen use efficiency.
Yeah.
So one of the concept is that if you can possibly if you can improve the activity of nitrate reductase, probably you have to use less fertilizer, and huge amount of fertilizer is used. Even if you can cut down 10-15% of that can save a lot of money.
That can save money. That can save the quality of soils, because you're not putting too much nitrogen fertilizer.
Yeah.
So that's a very important protein. And you mentioned the term yield. Yeah. We are talking about cereal crops and grains, right?
Yeah.
Rice, wheat,
right.
corn, etc.
Right.
And talking about wheat, of course, I wanted to mention the and you did your PhD around 80s, right?
Yes.
Yeah.
Yes.
So I just wanted to mention that Professor Norman Borlaug, he used to be at Texas A&M University, and he won the Nobel Peace Prize, of course, and he developed the dwarf and disease-resistant wheat that really made India self-sustainable in rice. And now I think India is the net exporter of wheat as well.
Yeah.
So that work was done. It's still, he was credited he is credited for having saved more than 1 billion people. And I think it won't not be a wrong statement to say that you and I are here because of a lot of that work, because really, really fed the people over there.
Yes, yes. There was a time when India was, very short on the production level of wheat, and India imported wheat from different countries, and a lot of that was not of very good quality.
Yeah.
But since there was very poor availability, people ended up consuming that.
Yeah, importing and gift also. I mean,
Yeah.
because we had a large population that needed to be fed.
Yeah, but now things have turned around, and now India is exporting wheat to many countries.
Yeah, yeah.
And so things have changed, and really that what you call green revolution in India, Borlaug contributed, and on the Indian side, Dr. Swaminathan
Yes.
contributed for that.
And for rice, Dr. Gurudev Kush.
Yes,
You know,
yes.
so I know that happened. I think this just also goes to show the work can be done anywhere in the world, but it can benefit the entire globe.
Yes.
So science has that type of reach beyond boundaries and
Yes.
beyond geographies. So talking about, of course, grains. I mean, this is the time when we needed food, but I wanted to pitch something for horticulture now. The big part is that we have focused a lot on meeting the calorie needs for the population. However, FAO just released a report on October 16th. Their report said that we are so far behind the goals we've set for nutrition and hunger. And malnutrition is not just a problem in the developing world. I mean, malnutrition is a challenge also in the developed parts of the world as well. So, do you reckon the second green revolution has to include horticultural crops?
Yes, definitely, because when you don't have anything, a lot of scarcity of food, then you want to eat anything.
Yeah.
But once you have options available, and still if you are not getting balanced food, then something has to be done about that. And so basically we have to improve the quality of food. It should be more nutritive, and if you have plenty of availability, but if you have some people, like part of population, they are not getting access to that food, that also is a problem. So we have to have better quality of fruits, and they have to be more affordable also. They should not.
Well, fruits, vegetables, nuts.
Yeah.
Well, so by some statistics, about 85% of farmers around the world are smallhold farmers.
Mm-hmm.
And I think when we talk about raising their economies, there are examples where growing horticultural crops, which are low volume but high value, compared to cereal crops, which are high volume but low value per acre.
Mm-hmm.
And I think we need to have a diverse production system even for smallhold farmers if they want to uplift their economic status through that. I mean, what do you think about that? I mean, diversifying that, because the focus has been so much on the cereals. It's like we're still hydroplaning on that concept of just meeting the calorie needs.
Yeah, so I think now people are more aware, that they need good nutrition, and at least in the market things are available. But I think there's kind of still, in some countries there's a population which can't afford, whatever is available.
Well, they can't afford to buy, but if
Yeah.
they are doing any subsistence farming,
Yeah.
they could also incorporate growing horticultural crops. I
Yes.
mean, there are examples where people would also have fruit trees. So permanent crops, which are nutritious, but also then you could process those. I think it's a cultural aspect as well.
Yeah,
I mean.
definitely, because processing is also very important, because when you have something like wheat, rice, you can store for a long time,
Yeah,
but fruits, they have limited shelf life. So like processing can kind of provides alternatives where processed food can be stored for a long time, and once the harvest is over, maybe for a year or so, people can still consume that kind of food.
Well, I want to go back to where we were at. You finished your PhD. So what was your journey from there on to becoming a professor?
Well, after PhD, that was the time I worked on, as I mentioned, I worked on phytochrome regulation of nitrate assimilation. But that was the time when molecular biology was growing up very fast, and my idea was also to get into molecular biology. So just after PhD, I spent some time with my PhD supervisor, and I requested him that I want to learn some molecular techniques, and there was a lab in JNU which was called DST Unit for Molecular Biology, and there Professor H.K. Das was working on improving, not just improving, like kind of incorporating nitrogen fixation in cereals. And so I got some experience of RNA isolation, northern blotting, and things like that. And then one of the faculty member of Yale University visited our department, and he advertised a position for postdoctoral candidate. And so several people from School of Life Sciences, were called for interview, and I got selected for that position, and that's how I started postdoctoral research at Yale University. But the area was not plants there, and my interest was to learn either way, whether it's plant, animal, any system.
Yeah.
So I learned basic molecular biology there, and my main focus was on study of DNA-protein interaction there, and so that's how I spent some time in Yale, and then he moved to Detroit, Wayne State University, Detroit. I also moved. I spent 2 years there, and the problem changed a little bit, and I worked on JC virus. This is again polyoma virus like SV40, and it causes a disease in brain which affects myelin sheath, and it's a complicated name: progressive multifocal leukoencephalopathy.
Okay.
So it's kind of disease which is there in silent condition in about 50% of the population.
Wow. Okay.
And when somebody is immunocompromised, it starts shooting
Flare up. Okay.
Yeah.
Okay.
So
just like strep throat.
yeah. So it basically affects brain. So then I studied, how this brain-specific expression is there for the disease, and I worked on the promoter of JC virus, and we found some brain-specific element which make it express in brain.
Okay.
So we saw expression in HeLa cell, we saw expression in brain cell, and compared, and we identified some negative cis-acting elements which were responsible for expression in brain cell, not in HeLa cell.
Wow. Okay.
Yeah.
Wow. Well, so you went. First of all, it's pretty cool and not surprising that light perception, or where plants sense light, would be connected with nitrogen, because as light gives the signal for growth, it obviously says I need to suck up more nitrogen to make more proteins.
Yes.
And then from plants you went to brains. Yeah, I mean this also showcases that the scientific knowledge also doesn't have any boundaries.
Yeah.
And we can learn from various disciplines, because sometimes, and most of the times, because of whatever reason, we become so siloed and learning only from what we know or where we are. But I think expanding those horizons can be good. And I recall we used to discuss, a lot of things, because I was doing signal transduction work, and we used to discuss calcium signaling is related to blood pressure signaling in people, in humans. And we used to learn from those ideas as well. So that just reminded me of that example as well.
Yeah, so after my postdoctoral work in US for 3 years, I went back to India and again joined Professor Sopory, and that was again postdoctoral position, and I worked there for 3 years.
And this was JNU, which is the Jawaharlal Nehru University, and Jawaharlal Nehru was the first Prime Minister of India, so it was named after.
Yeah. So I worked there again, and there I started my interest in epigenetics. So
you were doing epigenetics when it was not the cool piece of the science, right?
Yeah.
So, and just for our listeners, epigenetics is a modification of DNA without any changes in the letters of DNA.
Yes.
Just the chemical changes that can change its workings, right?
Yes. So when I joined Delhi University South Campus, so already we had a professor, Professor Khurana,
Yeah.
who was working on phytochrome.
Yeah.
So I was advised not to have a clash of the field, and since I had interest in epigenomics, I
Yeah.
started finding out role of methylated DNA binding proteins in plants. And at that time, there were some proteins known in pea, carrot, which bind to methylated DNA, but nothing more than that.
Yeah.
So I was exposed to a lot of presentations regarding this. There was a student in Professor Sopory's lab who was interested in methylation. So there's a person, Adrian Bird in UK, I think Scotland probably, and so he was the authority for study of methylated DNA binding proteins in mammalian system.
Okay.
So I requested him some clones of mammalian protein, and I wanted to find out whether plants also have got those proteins. So I did southern analysis.
Well, just
using those throws.
that's a very technical term, but basically
Yeah.
you were studying DNA,
Yeah.
trying to understand its structure.
Yeah.
Yeah.
So basically, then I kind of compared the sequence of mammalian protein in the Arabidopsis genome information, whatever was available by that time. Whole genome was not sequenced.
Yeah, I think that came out in 2000. So
Yeah.
this was
Yeah.
early 90s that you were working on?
I started, I think, 98, 97,
Wow, okay.
97, yeah.
Yeah.
Yeah. So basically, we found some homologous sequences in Arabidopsis,
Okay.
which was similar to mammalian protein, which is called MECP2. And so we found 2 hits, and we cloned the genes for both of those proteins. And when we started working, a publication appeared which kind of generated database for methylated DNA binding proteins or Arabidopsis. And there were 13 proteins which
Okay.
were listed. And our protein, which we started calling 1 and 2, they were 10 and 11 in that database.
Oh, okay.
So then we started, finding out the function of those proteins.
Yeah. And now we can sequence the DNA and actually in different methods to really find where that methyl group, CH3 group,
Yes.
or
Yes.
acetyl group
Yeah.
in the protein could be to modify DNA. And that is what really controls. And now we also know that it can be passed from one generation to the next.
Yeah.
It's
Yeah.
fascinating, at least in plants.
Yes.
There is evidence for that.
Yes.
That's amazing. So that was your postdoc. So the journey to professorship now.
Yeah, so I also worked on another project in collaboration with Professor Akhilesh Tyagi, there, my senior colleague, and
my
yeah,
PhD advisor.
Yeah,
Yes.
so
Both Dr. Khurana and Professor Tyagi.
yeah.
Yes, yes.
So that was the project for development of edible vaccine,
Yeah.
and
So that was a biotech project, plant molecular farming. This is like 2099.
yeah.
Wow.
So I think after 2-3 years of joining, I started
Yeah.
that project, and there, the concept was that kind of producing antigens in a way that people abide by vaccination. They don't hesitate, because when we were young, we used to get a lot of vaccination for like smallpox, cholera, and things like that, and that used to happen in the schools. And when it was announced that on so and so day vaccination would happen, people used to run away
From the needles, yeah.
yeah. So, like, if you express those genes encoding these antigen candidates in edible fruit, and tomato is one such fruit where you can express these genes, it's available year-round,
Yeah.
and so availability is good. So we decided that tomato can be that system where we can express such genes, and we did express few of the antigens of Vibrio cholerae, which is the causal bacteria for cholera.
Cholera, yeah. Dysentery.
Yeah.
Or
And so,
diarrhea, sorry.
Yeah. So, I mean, if you express gene coding for Vibrio cholerae, there is always concern that, it's kind of toxin and you can create problems. But, it has 2 parts. One part is called B subunit, which binds to gut. It's not toxic. Another part is A subunit, which binds to B subunit. and that is the one which is toxic. So what we did, we used B subunit as the antigen candidate. So idea is that if you produce antibody against that, then toxin will not be able to bind to the receptor.
Yeah.
There is a receptor in the gut for cholera toxin B
Yeah.
subunit, which is GM1 ganglioside.
Yeah.
So we express Vibrio cholerae antigen, this B subunit antigen. We also tried other, because there is a concept that this may not be enough, because it can block the binding of toxin, but it may not block the colonization of the bacteria. So amount can keep increasing. So there are some anti-colonization factor. So we also express the genes for those, and we had some experiments done in mice model systems, and we found that when we fed tomato expressing these antigens, mice, they could produce IgA antibodies against those antigens, and we compare, whether titer is similar to when you
titer, number of microbes, right?
Titer is basically how much of antibodies are being produced.
Okay.
So we found that it was in similar range compared to when you inject pure toxin.
Okay.
And this was toxin expressing in tomato.
In tomato, plant-based, yeah.
Yeah. So we could publish few papers in that subject, but this didn't go a step further.
Well, that's why I really wanted to come. I know that there was a big hype. This was late 90s, early 2000. There was a big hype being made by several scientists about edible vaccines.
Yeah.
Where did we miss the train on that, or bus? What came in the way of getting it to the people, to the masses? Because I know that, in a healthy world where there is, of course, herd protection, people think vaccines may or may not be important. But when you are in developing areas, developing parts of the world with heavily populated areas, where you can have this disease completely be avoided, like diarrhea through Vibrio, cholera.
Yeah.
So what really happened? Why didn't we get it to market?
I think I would attribute to two factors. One is that for immunization you require certain dose. So if you can't express at very high level in tomato, for example, if you need say 5 microgram of the antigen, if this 5 microgram is there in 10 gram, probably you can feed. But if this 5 microgram is there in 5 kilogram,
you can't eat 5 kilogram tomatoes.
So you can't feed that much of food.
Yeah.
So expression level was one bottleneck. Another bottleneck was that earlier it was thought that you can grow this kind of crops in the field and people can pluck and they can eat and they can get immunized. But in this system, dose is very important. So when you express something in plants, this expression level can change in different tissue. It can be different in upper tomato. It may be different. Lower ones, it can be different. Environmental condition can change. And dose is very important. Like if you provide a dose which is not appropriate, then instead of generating immune response, there can be immunotolerance. May not be that no response is generated. So dose becomes important. So then that means it requires some processing, and that makes it a little more complicated.
No, that's true. I know that now we don't talk about edible vaccines, but it still remains an interesting concept where if we could regulate the dosage, it could be easily administered to the masses if needed. So that was one part, and I just wanted to say, I know that there are, the whole area of molecular farming, of expressing pharmaceutical proteins or ingredient proteins or other chemicals in plants is still active, and there are companies and labs really standardizing a lot of that work as well. There is some potential there, but I think dosage would remain a challenge.
I think there are companies who are taking up this area, but things have changed a little bit. Like when the concept was started, one of the scientists of Boyce Thompson Institute, Dr. Charles Anjan, he promoted.
Actually, he was here as well at A&M too. So,
So he was thinking about banana at that time, because banana is part of baby food, and most of the vaccination is required in that age. So banana is not easy to transform, and protein level is not very high. So if any tissue has low amount of protein, then foreign protein that you are expressing is going to be a fraction of that level. And you require higher levels. So now focus for proteins of pharmaceutical interest has shifted to other tissues like seeds. Maize seed is one of the important target, and already some proteins have been produced. For example, avidin has been expressed in maize, and the natural source of avidin is egg.
What does avidin do, by the way?
Like its main use is in diagnostics, basically. So so now you can buy avidin express in maize from Sigma. It's there in Sigma catalog, and the cost is less than for the one where you use egg for isolating avidin. So maize express avidin is cheaper than avidin purified from the egg.
So that's a more sustainable way to really produce, because you're not really raising animals to do that as well. So that's, talk about sustainable. I just wanted to say that our department has a mission around sustainability, wellness, and food security. And I know one of your other projects that we would want to discuss about is food security. Everybody talks about that we need to grow 70% more food and 30% less land. But I think a lot of the conversation misses another major point. We lose 40% of our produce after it reaches the shelf. So some of your work in that space really talks about food security as well. I mean, sustainability is, of course, for us means making sure that we generate knowledge, train people who can go and add to the economy and profitability of our industries. Wellness for us is eating nutritious food. That's why I talked horticulture. And then also interacting with plants gives you mental well-being. And then finally, food security is not just, being able to produce food, but being able to get it to the masses, as well as the capability to grow that food. So let's talk a little bit about some of your recent work that you've done on, towards food security.
So I think, as you said, there are two ways to improve availability of fruits. One is by increasing yield. Another is by avoiding losses. Because of over-ripening, about 30% or a little more than 30% of the, I can talk about tomato, about 30% of the yield is lost because of over-ripening. Because when over-ripening happens, one thing is quality becomes non-desirable kind of thing. Then it also attracts a lot of infection, fungal and all, So we lose a lot of this.
So infection in tomato. I mean, basically the fruit starts rotting.
Fruit starts rotting. And then also, when fruits are not firm, then during transportation also there is loss.
More damage,
So I think we have to focus on this aspect also. And so I worked in this area. And so we wanted to create tomato which have better shelf life. And we...
What about flavor?
Flavor is also important. I know.
Because now we get these tomatoes that taste like cardboard,
So I think these tomatoes came in a period where, a lot of commercialization of tomato happened, like people wanted to send them to long distances. And these tomatoes are like little longish and kind of square, and they can be packed in crates very well.
The shape,
And they are firm, so more flesh is there, but juice is less and sourness is less. So in terms of taste, many people don't like, but they are suitable for certain application where you need more bulk. So these have become more popular.
What about their nutrition?
I mean, if you see, they are not very red also in many cases.
So they will be lacking in the pigment that's good for our eyes, right?
Also.
The lycopene.
So, so one way of improving shelf life is, we know that ethylene is the one which is responsible for ripening. So
I think a lot of people are aware of ethylene because bananas, when they come green, or if you put your banana with an unripe avocado. The banana releases ethylene, so you can ripen avocado or a mango or a papaya with that. So they naturally produce ethylene as a gas.
Yeah.
So that's the one you're talking about, the
Yeah.
ripening one. So
Yeah.
tomatoes do that as well, right?
Yeah, tomatoes also. Like we have fruits of two categories. One where, ethylene level goes up during ripening gradually. And then other categories where when ripening starts, there is a burst of ethylene. So there you have, heightened activity, heightened metabolism, increased rate of respiration, bursts of ethylene.
Yeah.
So ripening progresses fast in those.
And some of the
fruits.
ones that don't require ethylene, examples would be cherry, grapes, citrus, which do not, although citrus when exposed to ethylene can change color.
Yeah, many,
but they don't ripen.
They require very low level of kind of continuous presence of
Yeah.
low level of
But this
ethylene.
burst
Yeah,
is like
is-
specific for bananas, tomatoes,
for-
apples.
Yeah, which are called climacteric.
Climacteric,
fruits.
yeah.
So basically, our idea was, like there were already reports of trying certain, genes for improving shelf life. Long back, there was a variety developed by Calgene where polygalactourinase gene was silenced.
Yeah, what does this gene do? It strengthens the cell wall?
No, like when this is active, this is part of pectin basically.
Oh, pectin.
So,
Pectin is that jelly-like substance with which you make jams and jellies.
So basically,
yeah.
you have polygalactouronic acid, and this has carboxyl group, and so you can have two strands of this, and there can be calcium in between, so it forms jelly-like thing.
Okay, so that's what the jelly comes from, the jam jelly that we make. That's the pectin piece.
Yeah. So basically, there is also cross-linking, like methyl esterification of these groups.
Okay.
So when these are methylated, they can't get cross-link through calcium because COH is blocked. But when these are carboxyl group, the calcium can bridge and it can form network, mesh kind of thing.
So you need calcium also?
Yes.
Well, this is right. We need calcium for everything, but not only our bones, but also the integrity of these cell walls as well.
So the result of that study was that there was not much improvement in the shelf life. So now,
it logically makes sense. If you can keep the cells firmer or pectin more firmer, maybe it will, but it doesn't because there are overriding aspects to this.
Yeah. So in that study, there was not much advantage in terms of shelf life, but probably pulp increased a little bit or some minor changes were there. So that was not very viable commercially. So basically, the idea is that if you block ethylene action, you can delay ripening. You can create firmer fruits.
Tomatoes.
Yeah.
Okay.
But then there is a kind of penalty for good pigment, which is lycopene. So you can have firm tomato, but they would look orange or not
deep red as they should be.
Not deep red as they should be.
Well, that's why people still like the heirloom tomatoes, which are deep red. They have all the flavors in them, but obviously they don't last more than 2 to 3 days.
Yeah. So my idea in this, like the way I approach this problem, is that like if you analyze ethylene signal transduction, you have ethylene, it binds to its receptor, and then there is a kinase CTR. Then downstream to that, there are EN2 like genes are there, 1, 2, 3, 4, like that, and then there are ERFs.
Ethylene response factors.
Ethylene response factor, and these are like around 100 in number.
Okay.
So my idea is that if you block ethylene action at very high level, then all aspects of ripening are going to be
So it's like a chain reaction, right? You start one gene starts affecting 2, 2 starts affecting 6 and 8, and so it's like a cascade.
And ultimately it's
Hundreds of genes,
So now these ERFs can be more specific. Like if you play with the ERF, because ERFs are of both kinds, some positively regulate ripening, some inhibit ripening.
Yeah.
So if you can find if you can characterize ERF, that if you find ERF which is only going to affect pigment development, then either by enhancing the expression of this or by lowering the expression, depending on whether it's inhibitory or stimulatory, you can increase amount of pigment. And same way, if you find ERF which is specific to cell wall degradation, then by modulating the expression of that ERF, you can try to generate firmer ERF. And there are some success stories, like one study came from Professor Ashish Datta's lab at NIPGR, Delhi, and they use two enzymes of cell wall.
Yeah.
I think manosidase and hexaminidase, I think two of such enzyme, and they got tomato which were red, deep red, and they were found for long time.
Good, okay.
So they could improve shelf life, and there was no penalty on red pigment, which is of good nutrition value. So some success stories have been there, and we have characterized some ERFs. We recently we published a paper in Plant Physiology where we found that there is a ERF which is involved in control of ripening, and its action is via ARF. This ERF binds to the promoter of ARF.
So ARF is the auxin response factor.
Yeah.
So both ethylene and auxin are what we call them at phytohormones.
Yeah.
Hormone by definition is produced elsewhere, but acts, moves to move else and makes a action. So auxin is produced in the meristematic tissues, in the apical tips, and then it gets distributed. But ethylene is produced in the cell anywhere, but it's gaseous.
It's gaseous.
But auxin is a chemical. So you're saying there's an interaction between between ethylene and auxin, which is, we know that they kind of counteract most of the time.
Yeah. So we characterize the way this ERF works in promoting the fruit, and we found that it interacts with the ARF, and that's how it promotes the ripening.
So is there a name for this ethylene response factor or ERF?
This ERF is called ERF D7.
Okay, just a name. Okay.
Yeah.
Nothing creative.
Yeah, I mean,
it's just numbering
name
system.
doesn't indicate what is the role of this.
Yeah, I know we're boring. Scientists can be boring sometimes. We just name things like that.
Yeah.
Okay. And so, what does it do? So when it interacts with auxin, how does it change the quality of the fruit or shelf life?
Well, it's it affects all aspects of ripening.
Yeah.
Like it affects softening as well as pigment development. So that means if you silence either of these, both aspects, softening as well as pigment, are going to be affected.
So now, are there tomatoes that can be commercialized with this? Or before we go there, how much shelf life was increased with this? How many days?
No, basically, I think it was difference of, 4 or 5 days. Not very serious.
But 4 or 5 days means a lot.
Yeah.
But if you have a flavorful tomato that is deep red, nutritious, has some flavor and is firm.
But that's the problem, because with this ERF you can't delineate two aspects.
Okay. You can't separate the
pigment from softening.
So the pigment was still somewhat orange.
Yeah.
But it was firmer.
Yeah.
Okay. But 4 to 5 days matters, right? I mean, you can stay on the shelf longer.
Yeah, but 4 or 5 days, if quality of tomato is not affected, then it's good. But if quality also goes poor, then for a fundamental study is good, but I think to go into commercial production probably is not suitable.
So people could look at the variations of this gene in the tomato germplasm or collection and see if they can use that as a parent for breeding. And now we can do gene editing, where we can really go and simulate the natural mutations that happen.
Yes, I think maybe one of the possible idea can be that we characterize few of ERFs, 4, 5, but there are hundreds. So if more of the ERFs are characterized, maybe we can find one ERF which is affecting only pigment.
Only pigment, okay.
And then playing with that can allow development of good kind of tomato, which will have high level of lycopene and also higher shelf life.
Is somebody continuing this work?
Not in my group because after the retirement I left the lab, and currently there is nobody in the department who has taken over tomato as part of research.
But since you published it, maybe there are other groups in the
yeah,
world
some might
that
be
might be working on that. So that's
yes.
very interesting as well. So I also read about your story. We heard your seminar on methyl glyoxalase. Where does glyoxalase fit in? And also, if you don't mind sharing, what is that chemical? Because that also enhances shelf life, right? You figure out a mechanism for ripening. So
shelf life, not exactly, but what we have shown is that this is a kind of novel pathway which was not known earlier to affect fruit ripening. Because mostly people tried, finding role of different hormones, ethylene, auxin, ABA, and jasmonic acid. They have some role. And recently, there is also a peptidal hormone in plants, which are not too many. It's kind of phosphokine. So it's kind of 5 amino acid peptide, and there is a receptor, phosphokine receptor, where it binds, and then this receptor interacts with DREB, and it phosphorylates DREB, and this DREB promotes fruit ripening. So I mean, this is not something which is very well studied.
What is DREB?
This is desiccation response element
binding protein. So desiccation is just lack of water. And
Yeah. So what I'm trying to say, this is something which is not very
well studied.
Well studied. Similarly, this glyoxalase and methyl glyoxal pathway, first information about the presence of glyoxalase in plants was uncovered by my Ph.D. supervisor, Professor Sopory. And earlier than that, it was known that it is there in animal system, mammalian system, and there was some correlation established between metastatic activity and increase in glyoxalase activity. They may not be cause and effect, but there was association observed. So Professor Sopory's group also, checked whether fast-growing cells have more glyoxalase activity, and they also found some correlation. But later on, because at that time there was not solid evidence of the presence of glyoxalase in plants, so his group purified the protein, acid, cloned the gene. Once they cloned the gene, they tried to find function of that. They overexpress in different crops. And when they overexpressed, they found that those crops became stress tolerant, tolerant against salt stress especially.
So glyoxalase, by the name, indicates it's an enzyme.
Yes.
It acts on which chemical?
Yeah. So basically, there is a chemical called methyl glyoxal.
Methyl glyoxal.
Yeah. So this comes
So that is what accumulates in the plant.
That's what accumulates, and this comes from a side reaction of glycolytic enzyme.
Which would be?
And there is a step in glycolases where you have production of glucose molecule is converted to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So then this dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate by triose phosphate isomerase. So this enzyme triose phosphate isomerase, it has a kind of loop which prevents side activity. So the hypothesis is that when you have some kind of stress, maybe proteins are misfolded or they're not in native conformation, this loop doesn't work, and then this activity, same activity, generates instead of glyceraldehyde 3-phosphate methyl glyoxal.
So in a fruit has sugars, sucrose. Sucrose breaks down to glucose and fructose, and then glucose gets further metabolized or broken down. And so as it is being broken down, there could be there is a production of methyl glyoxal on the side.
Yeah.
And that is just happening during ripening, and it gives us the sweetness, and the sugar gives us the sweetness, the glucose. But
so basically, like if you see green fruits, there is a general correlation that when you have more level of glyceraldehyde 3-phosphate or dihydroxyacetone phosphate, these 3 carbon sugars, then you have more level of methylglycol.
okay.
So that means what we try to find out whether methylglycol level changes during ripening. So we checked the levels in green fruit, red fruit, and we found that when fruits go towards red color, there is decrease in methylglycol level.
When it turns red,
Yeah.
Okay. So that's very interesting. So it's part of the ripening process then.
Yeah.
So I have a question.
Yeah.
If you apply methylglycol to a green fruit, will it stay green longer?
Yeah, that's what happens.
Oh, cool.
So if you apply methylglycol, it inhibits the ripening. And you increase the concentration, stronger is the inhibition.
So is that possible for all fruits? Or is it very specific to tomatoes?
Because our more, like we did more work on this, and what we find that this Glyoxalase system is what to say. I was trying to hit a point.
Is like a universal system or no?
So basically, what happens is that as the ripening progresses, there is change in methylglycol because earlier its level is high,
And then starts lowering.
and then starts lowering down, and this Glyoxalase and this lowering, and then we try to find out why this level comes down.
Okay.
And we found that there is corresponding increase in the activity of Glyoxalase. So we tried to find out, characterize gene family of Glyoxalase, and we found some members which were showing fruit-specific expression.
Okay. So you're saying that as the glyoxalase enzyme goes up, it starts breaking down methylglyoxal,
Yes.
and that allows for the fruit to ripen.
Yes.
And this is happening in a fruit called tomato,
Yes.
which we have traditionally thought ripens because of ethylene alone.
Yeah.
But this is
Yeah.
also a side reaction that is happening
Yeah.
during the ripening process.
But still ethylene is a
Involved,
player.
of course,
Yeah.
of course.
So, and we try to prove this point, it was just a correlation. So then we silence Glyoxalase enzyme, and then we saw what happens.
Okay, so you shut down the enzyme using genetic engineering.
Yeah.
Antisense?
We
RNA.
use microRNA based silencing.
MicroRNA based silencing,
Yes.
okay.
Yeah. And when we did that, it inhibited the ripening.
Yes.
And it increased the level of methylglycol because that kept accumulating.
Yeah.
And so now question comes, what does methylglycol do?
Yeah.
How it blocks ripening, and the possible reasons are, what we know from literature, and we also documented in our study that methylglycol can bind to many macromolecules like proteins, lipids, nucleic acid. In the proteins, this can form adducts, which are called advanced glycation end product. So this can affect lysine, arginine, cysteine.
These are the amino acids.
These are the amino acids which are targeted, and these glycation products basically can change the property of protein. And we detected this, like we did Western blotting. We isolated the proteins, and there is a way of detecting whether this glycation has happened, and there is antibody for methylglycol which can be used, and this will show. So when we had situation where level of methylglycol was more, we saw more bands for glycation of the proteins. So
it was modifying other proteins as well.
Yeah. So
because it's gotten so high. So okay.
Yeah. So basically,
is it toxic, methylglyoxal?
I mean, toxic in the sense,
not to people, but metabolically toxic.
Yeah,
that it is stopping reactions, not toxic to us.
Yeah,
But,
that's
okay.
what it is.
Okay.
So it is affecting a lot of proteins because
very
it is reported that many stones are also modified because of this.
And that's where epigenetics comes back.
Yeah.
So it connects with the modification of DNA without changing the letters, right?
Yeah. So it can modify histone. It can also modify guanine. So it can affect nucleic acid also.
Okay. Also, it can chemically change.
Yeah, it can chemically
DNA
change.
letters. Okay.
Yeah, yeah.
Oh, wow.
So this is kind of a molecule which can create a lot of havocs.
I'm curious to know. So we talked earlier about climacteric fruit and non-climacteric. So this is obviously happening in tomato, climacteric.
Yeah.
Any evidence of a similar mechanism in cherries or grapes, limes, which are non-climacteric?
I think we have checked only in
tomato.
Because
there is a whole idea of how do those fruits ripen.
And no other paper on role of Glyoxalase in ripening has appeared. So
okay, so the field is open, right? Because
Yes.
I work in that space. So I'm very curious about,
Yes.
because there's a whole hypothesis that fruits they're all related.
Yeah.
So there are some plums which are both climacteric, like mutants, and non-climacteric in the same system. You can also say peach and cherry, they are cousins. But cherry is non-climacteric, and peaches are climacteric. So there could be some similar so it's worth exploring. I think that's a very interesting topic.
So I was going to say one more thing. When we block Glyoxalase enzyme, this ripening is inhibited because we see pigment is not developing. So we try to find out where is the block, because it seems, and we check ethylene. Ethylene level is compromised.
Okay, so it's reduced.
Yeah.
Okay.
But then which step of ethylene biosynthesis is affected? So we try to find out.
Yeah.
So we checked whether like precursor, like methionine.
Methionine would be the precursor.
So we checked aspartate family amino acid, including methionine, and we found that methionine is showing fruit-specific increase.
Oh.
So when we silenced the Glyoxalase and that inhibited the ripening, we tried to supply methionine. So in wild-type tomato, it improved little bit of color development, but when we had a silence line, it didn't work.
So where the glyoxalase enzyme is not working, methionine was not able to ripen the fruit. But in wild-type, which is natural fruit, you provide methionine, which helps in increase of ethylene, and it ripened the fruit then. Made it more, the pigment became redder.
So then we checked, methionine couldn't supplement. Then we tried ACC.
Okay.
So we used this tomato where...
ACC is a precursor of ethylene.
Yes. So when we supplied ACC, that could, even for the Glyoxalase silence line, it could improve the pigment.
Oh, okay.
So that means when Glyoxalase is knocked down, methyl glycol level is high, tomato fruits are not producing ACC.
Very interesting. So this could really help, going back to food security. This is another piece of knowledge that has come in that can help, and one could generate mutants in this pathway to really find out nutritious food, but also firmer fruits as well. I know earlier you called tomato as a fruit. Some might argue that's a vegetable.
Yes.
Botanists understand. It's the used term, vegetable, but it is a fruit, of course.
For a layman, it's a vegetable crop.
Yes. No, wonderful. And also, I think, so when we were in the late 90s, Professor Akhilesh Tyagi initiated the rice genome project. I was still in the lab over there with him. And I think we were, just after that, there was also an international consortium, and your lab took the lead on the sequencing of the tomato genome. Can you share that experience? And
So basically, when talk of sequencing of tomato genome started, then Professor Tyagi was already part of rice genome sequencing. So he joined, and he asked me, because I had interest in tomato, would you participate in this program? So I agreed. So I also became one of the investigators in that program. So India was nominated for sequencing of chromosome 5. So it was sequenced at 3 places in IRI, Indian Agriculture Research Institute, our department, and in National Institute of Plant Genome Research, NIPGR.
Okay.
So we decided, we've distributed the portions.
So I want to just ask one question about the tomato genome. I mean, just as, or make a comment, as the human genome project happened, where that was sequenced, it was like 4.3 billion DNA letters.
Mm-hmm.
How many letters does tomato have?
Tomato, I don't remember exactly, but I think it's about 900.
I mean, it doesn't have to be exact.
MB
I mean,
or something.
So
like
it's in the millions. So 900 million bases,
Yeah.
much smaller. But just the way when we've sequenced the human genome, now we are finding genes related to different diseases, or what changes in DNA causes certain diseases. So we're getting all this knowledge. Do you think with the tomato genome now, I think now we have sequenced more than
Yeah.
100 tomato genomes,
Yeah,
right?
now a lot of cultivars have been sequenced. So I think it's going to help in molecular breeding great deal, because with sequencing of so many cultivars, physical markers must be saturated probably, and now if you want to do molecular breeding, you have more resources because of genome sequencing.
And knowing the DNA print, like blueprint, we can also create mutations through gene editing
Yeah, and
to really rapidly improve nutrition and the ripening and keep the fruit firm.
yeah, and this gene editing is also providing another big advantage, because so far, as you know, in India, I think only 2 crops have been approved. One is the one which is commercial, BT cotton,
Yeah.
and another is brassica, which is developed by Professor Pentel of genetics department, and there's a lot of resistance, and it involves a lot of paperwork, lot of trials, and huge investment of money for release of GM variety in India. But now government has come out with the regulation that if you do genome editing by CRISPR-Cas system, then these lines would be considered equivalent to natural mutants.
Yeah, because you can, after the mutation is done,
you can segregate
out.
the
But that's a really
Cas.
big thing. But I think having this knowledge can finally help us reach the goal of a flavorful tomato
Yes.
that is healthy
Yes.
and can meet the nutritional needs, and people can enjoy it over and over again.
Yes, sure. I mean, this will not only tomato, I mean, in any crop, because right now, there are many labs which have generated transgenic plants, different plants. But because of these regulations, they didn't see light of the day. But with the change in regulation, now already, different groups have started using CRISPR-Cas system.
Yeah.
So whatever they achieved by conventional methods, like regular transformation, now those plants, once they are coming through CRISPR-Cas, probably regulatory approvals would be easier to come by.
Yeah.
And we'll see probably more GM crops, although still there is a big lobby of environmental, NGOs who are opposing, and they are also misleading farmers into believing that if you start growing GM crop, and these are owned by big companies, and so you won't be able to multiply your own seeds, and they have some kind of terminated technology, and
which is all.
Yeah.
It was all fiction, and I
Yeah.
think unfortunately.
So
Yeah.
and farmers are a big political lobby, so even government is not bold enough to
Well, I think it depends where you are in the world. I think recently the purple tomato, which has very high levels of anthocyanin and nutritional components, has been commercialized, and that is actually available. It is becoming available for people. So that's very interesting. So you had a very distinguished career, Professor Sharma, and so besides these scientific contributions, I think you made another contribution, a massive one, training the next generation of people. So I don't want to make counts, but you've had several masters and PhD students.
Yeah.
A specific number in your mind, or?
I think it's close to 15.
Wow,
15
okay.
PhDs and
Wonderful.
some MPhil students. And we also take master student every year, like 1 or 2. So maybe 20 around master students who went through the training. So yeah, that's our job, to train
So you've created
students.
the next generation. We were talking about tomatoes next generation as well.
Yeah.
So you've done that as well. So really congratulations on a distinguished career as well. And I just wanted to say, do you have as you have crossed this journey, now you're the other part of it, or of course you're starting, the journey continues. But my question is, do you have any message for future scientists or students?
Yeah, I think message is that I think here I remember something after I passed my MSc program. We had a distinguished professor in our department at Merit, Professor V. Puri.
Mm-hmm.
He was famous morphologist, and he came to the department and gave a talk to fresher students who were coming for the next batch of MSc. And he said, people, when they choose career options, they go by, what neighbors are saying, what friends are saying, which field is important, which is not important, which is going to fetch you the job, and things like that. These are the questions in the mind of students when picking up certain area. So he said, if you are average student, in any area, you will struggle.
Yeah.
But if you are much above average, whatever may be the area, you will succeed. So I think message for students from my side is that, that they should do something in which they have passion and where, they can put their heart and soul. And if they, pursue their goal and whatever it may be the area, they would be more successful. So what matters most is what you do, you should have passion for that. Field doesn't matter, you can succeed in any field. You have potential of doing that.
That's amazing. Well, again, thank you for mentoring so many people, including me as well. Thank you.
Welcome.
And again, congratulations, and thank you for being here with us today.
Thank you. It was nice interacting with you, Amit.
After many years, yes.
Yeah.
Well, thank you. We hope you enjoyed this episode of Horticultural Innovators. Subscribe if you want to learn more and spread the good word. Thanks and Gigam.